Assalamualaikum and a very good day to the respected panels and to the audience. My name is Shazasha binti Lakman from AS257. The title of my presentation today is Microalgae as Antimicrobial and Antibiofilm Agents, a review. Did you know that biofilm formation is responsible for 65% of microbial infection and 80% of chronic infection according to National Institute of Health? Medical devices such as intravenous catheter and urinary catheter provide an ideal surface for biofilm formation. Moreover, dental plaque is the result of biofilm forming activities by pathogenic bacteria that form on teeth and also associated with most of oral diseases. Once biofilm has formed, antimicrobial and immune response are ineffective in eradicating the infection. According to the World Health Organization (WHO), the emergence of multi-drug resistant strain is a major public health concern. Moreover, WHO added that the misuse and overuse of antimicrobials are the main drivers in the development of drug-resistant pathogens. To address this issue, WHO published a list of antibiotic-resistant priority pathogens. In order to seek for new bioactive compounds as antimicrobial and antibiofilm agents, the pharmaceutical industry has shifted attention to alternate sources such as marine life. In this case, the marine life, which is known as microalgae, both eukaryotic and prokaryotic, have been found to produce intracellular and extracellular metabolites with variety of biological activities. Hence, this review has highlighted the potential of microalgae as a source of bioactive compounds with pharmaceutical properties since they have been found to exhibit antibacterial, antifungal, antiviral, and antibiofilm activity. Moreover, microalgae can appear in green, red, golden brown, and blue-green depending on its pigment composition. Before I go further with this presentation, I would like to share a video titled Discover What Microalgae Are in Just 60 Seconds. Let's take a look of the video in the following slide.
What do you really know about microalgae? Microalgae are microorganisms that first appeared on Earth around 3,000 millions of years ago and created the atmosphere as we know it today. They are the first organisms that performed oxygenic photosynthesis and are therefore the origin of all known plant-based life. They are the reason why we have oxygen on Earth. And still today, microalgae living in the oceans provide us with more than half of the oxygen we have. Also known as phytoplankton, they are the first link in the aquatic food chain. However, we have not paid much attention to them until now, because we now know that they have great potential to improve our lives in many ways. We not only owe microalgae life as we know it, but also the improvement of our lives in the coming years. They are our origin, but they are also our future.
I hope the video that I have shared to you just now will give you more knowledge about microalgae. Next, some of the problems encountered are the rapid spread of multi-drug resistant strain, also known as MDR, cause infections that are not treatable with existing antimicrobial drugs. Next, the lack of new classes of antimicrobial and antibiofilm compounds worsen the MDR global threat. Moreover, the use of synthetic antimicrobial drugs causes consumers to experience side effects. Now, I will talk about the importance of this study. First, the study will be significant to pharmaceutical industry because the discovery of the bioactive compound produced by microalgae will be a valuable resource in the fight against antimicrobial resistance. Antimicrobial compound derived from microalgae can be used to replace the use of existing pharmaceutical drugs that no longer work to treat infection caused by bacteria, fungal, and viruses. Moreover, this study is important for biotechnology industry because this is a green approach of producing antimicrobial compound from natural sources. Moreover, this review will serve as a reference for future researchers because it provides insight into existing research and identifies needed research, allowing future researchers to add more value, particularly in the search for bioactive compound in microalgae. Now, moving on for the literature part, I would like to highlight about various biological activities of microalgae and cyanobacteria. The first important topic that I would like to highlight is antibacterial activity of microalgae. The antibacterial effectiveness of a compound varies based on the gram type it encounters. Gram-negative bacteria are more resistant to a variety of antibiotics than gram-positive bacteria because gram-negative bacteria have an impenetrable cell wall called lipopolysaccharide that restricts extracellular compounds from penetrating their cell. Moreover, a study reported that antimicrobial effects have been attributed to phycobilioprotein, the light harvesting protein found in microalgae, which are responsible for the microalgae's ability to absorb light. For example, Porphyridium erigeneum produce phycotannin, while Propyridium crantum produce phycoerythrin, which found to inhibit the growth of Staphylococcus aureus. The next important literature is the antifungal activity of microalgae. The antifungal properties of microalgae has been associated with cyclic lipopeptides called hexylidine. A study conducted revealed that Anabaena species and Nostoc species produced the antifungal bioactive compound called hexylidine, which inhibited Candida albicans and Aspergillus flavus growth. The mechanism by which antifungal cyclic lipopeptide from microalgae works is mainly described by membrane disruption. Moreover, I would like to explain the antibiofilm activity of microalgae. Exopolysaccharides (EPS) production is part of the biofilm maturation process, in which the biofilm matrix gradually builds up and larger bacterial aggregate known as tower form. At this point, the bacterial cells begin secreting more EPS that enclose them, thereby stabilizing the biofilm network and guarding themselves from antimicrobial agents. Another important area of research associated with biofilm formation is oral health. Streptococcus mutant and Lactobacillus species are the most common bacteria linked to tooth decay and other oral diseases because of their biofilm forming activities. A study reported that ethanolic extract of the green microalgae Corella vulgaris and Dunaliella salina may facilitate in reducing biofilm formation. Scanning electron microscopy revealed that antibiofilm compound reduce microcolony formation and structural changes in exopolysaccharide EPS, limiting growth and virulent properties in Streptococcus mutant. In this slide, Table 2, 3, 4, and 5 summarize the valuable antibacterial, antifungal, antiviral, and antibiofilm compound that has been reported from previous studies respectively. After reviewing most of the published papers related to antimicrobial and antibiofilm of microalgae, there are some research gaps that has been identified. The first one is unexplored strain of microalgae. Based on biodiversity study, more than 50,000 species of microalgae have been estimated to exist, but to date, only 30,000 have been documented. Moreover, the low yield of microalgae valuable compounds, the lack of strain robustness, or low productivity is usually the reason these strains have not yet achieved commercial scale production. Next, lack of studies about antibiofilm activities of microalgae or its extract, particularly its mechanism of action and toxicity of antibiofilm compound produced by microalgae. For suggestion and recommendation, future research should concentrate on searching for optimal and industrial robust microalgae strains that are able to produce target and desired molecules. Furthermore, methods for improving yield, activity, and stability in the development of microbial compounds to meet industrial needs must be developed in order to achieve the best production result. Besides, studies on the antibiofilm activity, mechanism of action, and toxicity of antibiofilm compound produced by microalgae are urgently needed. Before I end the presentation, I would like to express my gratitude to Dr. Muhammad Taufik Mat Jalil, my supervisor, for his great advice and love support and help me in the completing this review paper and this presentation. Last but not least, here are the list of references that I use, and the list goes on until the next 3 following slides. That's all from me. Thank you for listening.